Hello students, I am back with yet another video of Nutrition in Animals chapter 2. So we are discussing about the small intestine and the different parts of the small intestine. We are also discussing about the secretions that the small intestine is receiving from other organs. So next we are going to discuss about pancreas today. So let us discuss about pancreas. So pancreas is a large gland. It is involved in the digestive process, but it is located outside the GI tract. As I have told you in the introduction also that there are some accessory glands which are involved indirectly in the digestive process. They are not involved directly. That is, food will not pass into these organs, and pancreas and liver are some of them. So it is composed of both exocrine and endocrine functions. It is about 15 to 25 centimeters in length and the weight may vary from 60 to 100 grams. So this is about the introduction to the pancreas. Now let us discuss about the secretions of the pancreas. So the pancreas secretes pancreatic juice. Okay. So the pancreas secretes pancreatic juice, and this pancreatic juice contains pancreatic amylase, trypsin and lipase. Let us now discuss the action of these enzymes one by one. So you can see here we all know that amylases are helping in the digestion of starch or carbohydrates into maltose, whereas proteins and polypeptides are digested by trypsin into peptides, whereas lipases will digest the fat molecules into fatty acids and glycerols. So these are the action or the role of the enzymes that the pancreatic juice secrete. So the pancreatic juice again will help in the partial digestion. Again, the pancreatic juice are not helping in the complete digestion of the food, but here too they are helping in the partial digestion of the food. So as I had told you earlier too, that there is a common duct through which the bile and the pancreatic juice will flow into the duodenum. So here too the same thing is being shown. Now let us see what happens in the ileum. So the wall of the ileum has intestinal glands that produce intestinal juice. So let us see what are the functions and what are the different intestinal juices. So the different intestinal juices includes peptidase, lipase, maltase, sucrase and lactase. So these are the different intestinal juices. Now as you can see along with the names you can recognize what the functions of these enzymes will be. So peptidase will act on the peptides resulting in the formation of amino acids. That is, the peptides that earlier was formed due to the action of the due to the action of the pancreatic juices will now break down into amino acids. Lipase will act on the lipids and break them down into fatty acids and glycerols, whereas maltase will act on the maltose and will break it down into glucose molecules. Sucrase will act on the sucrose molecules, breaking it down into glucose and fructose, whereas lactase will act on lactose, breaking the lactose down into glucose and galactose. So finally here you can see is that complete digestion of the food takes place with the or by the action of the different intestinal juices. That's why I told you that the complete digestion of the food will take place in the small intestine only, and here you can see which part of the small intestine is involved in the complete digestion of the food. That is, ileum is involved in the complete digestion of the food. Now what we have to remember is that first of all, talking of how proteins have broken down. So proteins have broken down into polypeptides first of all by the action of pepsin. Then the polypeptides have broken down into peptides by the action of trypsin enzyme. Then the peptidase enzyme has broken these peptides into the smallest units of the proteins, that is the amino acids. So this is how the proteins are breaking down into its simplest components in our body or in the small intestine. Now let us discuss the digestion of the starch. So starch first of all breaks down with the help of salivary amylase in our mouth into maltose. Then the pancreatic amylase will break this down along with the maltase into glucose. So this is how proteins and the starch is breaking down completely in our body, that is in the small intestine. Now we are going to talk of the next process, that is absorption. Now digestion is complete in ileum. What happens to the digested food now? So we all know that the digested food now has to be absorbed. So now the absorption takes place in the ileum, and the ileum has numerous finger-like projections which are known as villi on its inner walls. So here you can see a model of villi is shown here. So such numerous on the inner walls itself, numerous finger-like projections will be present. What is the role of these finger-like projections? So first we need to know what is the structure of this finger-like projections. So these finger-like projections have numerous capillaries. What are capillaries? Very thin blood vessels are known as capillaries. So this villi are rich in capillaries, so they increase the surface area for the absorption of the digested food. So let us see the detailed structure of the villi. So you can see one cell thick layer is present, then rich supply of capillaries is present which allows the transport of nutrients to different parts. Then the outer layer is just one cell thick so that rapid diffusion of nutrients can take place. Lacteal, a part of lymphatic system which allows absorption of fats. So this is how the villi is adapted for the absorption, that is the outer layer is very thin due to which the transport of nutrients can take place. It is richly supplied with blood capillaries due to which more and more digested food can be absorbed. So this is how the villi is adapted for absorption. Now ileum is, as I have told you, ileum is adapted to absorb digested food because of the presence of the villi. The surface area for the absorption of food is increased. Folded walls are also there. Villi and microvilli are present. So here again you can see ileum is very long. More time for digestion takes place. A greater surface area for absorption. Villi increases surface area for absorption. Villi is again divided into microvilli, increases surface area for absorption. Epithelium is very thin, that is the outer covering is very thin, so that the soluble products of digestion can pass through this layer very easily, and dense capillary of network is present so that the digested food can be removed from the small intestine and can be sent to the part where it is needed. So this is how the ileum is adapted for its function, that is absorption. So students, with this we have finished the small intestine. Now we'll be talking of the large intestine. So large intestine can be divided into cecum, appendix, rectum, colon and anus. So these are the different parts of the large intestine. I am repeating again: cecum, appendix, colon and anus. So now let us see what is the role of this large intestine. So large intestine consists of the materials that are present in the large intestine will mainly be containing water, mucus, dead cells and undigested matter. Most of the undigested matter that is present in the food is cellulose. Why is this so? Because in our body we don't have any enzyme to digest cellulose. Now what is the function of the colon? The colon helps in the absorption of water. So the water that will be present in the undigested food will be absorbed in the colon area, whereas rectum stores the feces before the feces, that is the undigested matter, will be expelled through anus. So this part, that is this large intestine, is involved in the process of egestion. That is, it is involved in the process of releasing the undigested matter from our body, that is egestion or defecation, the process of removing feces. Now what are the cecum and appendix? So it is a vestigial organ in humans which has no or small function, and it is large in herbivores. The both cecum and appendix are large in herbivores. We'll talk about this in ruminants. Now what is the assimilation of digested food? What happens in the process of assimilation? We have seen all the different steps. We have seen ingestion, we have seen digestion, we have seen absorption. Now we have come to assimilation. We have also discussed about egestion. Now the part that is left is assimilation. So the glucose is used during the process of respiration, and it is stored in the form of glycogen or fat. Fats help in respiration and also to build the cell structure as a store of energy. Amino acids, of course, they'll help in the buildup of proteins for cell structures and also for enzymes. They cannot be stored but are deaminated and they can be used in respiration. So the deamination can take place by two different ways. That is, amino acids is broken down. This process is only known as deamination first of all. So part with nitrogen forms ammonia, which will then form urea and it will be released out, whereas the rest is used for the production of energy. So this is about the deamination. So students, with this we have finished the human digestive system. I suppose you all have understood whatever we have seen till now. So thank you so much students once again for listening carefully and attending to the videos. Thank you so much everyone.